endemic wetland.
Thanks, James. All right, so this talk is one component of my dissertation that's kind of looking at the community, size structure of the community as a whole. And this one is specifically focusing on the small common fishes of the Everglades. So different processes act on different individuals and communities, and these processes focus obviously on the individual, and this scales up through the population, the metapopulation structure, and then finally up through communities. And a big part of this is environmental filtering, abiotic and biotic factors that affect where animals live, or organisms in general. And another big part is dispersal. Animals moving around, how well they can navigate through systems, are there avoidance of predation, things along those lines. And when you scale this up, that's how you really get meta-community function, and then beta diversity. And by beta diversity, I mean the differences in community structure between two different areas or a whole suite of areas. And this is a great example here of a study about 10 years ago looking at zooplankton communities in a series of interconnected ponds. That just shows both how filtering and dispersal can play a role as these ponds become connected at certain times of the year. And so these different processes can change over the course of a day. Think as an animal is just patrolling its territory. They can change over the course of a season, say snow geese migrating from Texas back up to Hudson Bay, or then over longer periods, like long-term dispersal where an animal moves and stays in that area. And so these conditions that affect how animals move are strongly linked to the environment. Think deep snow, presence of water. And these different conditions don't affect an animal just based evenly throughout its life. As animals grow larger, different threats of predation or nutrient requirements change, how these dispersal strategies change, and how environmental filtering plays a shifting role. And a big part of this dispersal is the role that the landscape plays. So resistance to landscape falls under three main categories. That's the physiological cost of an animal's movement, the reduction in survival due to that movement, and then just how willing an animal is to move through the landscape in the first place. And so thinking about the role of landscape structure and environmental filtering, I'm looking at a small fish movement in the Florida Everglades. Most of you are probably pretty familiar with this area, but it's a subtropical karstic wetland that basically flows from Lake Okeechobee all the way down through Florida Bay. It has a really strong seasonal rainfall pattern where 60–80% of the water falls between March and October or so. And it's just a shallow marsh. So any animals that are living in these deep water habitats, deep sloughs or canals, reside during the dry season in these areas. As the water levels go up, the animals move out into these shallow areas, and then as the water levels start to recede in October, they either make the decision to move back to these deep water habitats or risk desiccation or get eaten by wading birds. And this is just a great example of my study areas where you have the onset of the rainy season, the water level goes up, and then this was a particularly dry year where the marsh completely dried, which only happens every now and then. Water levels rapidly increase when animals start to move back, and then this is kind of a more typical year where the water levels might decrease to about ½ a meter or so. And so my question is, how do these movement strategies change based on landscape structure? And by landscape structure, I mean both the presence of or proximity to a deep water habitat, and then also the change in water level itself, which I consider part of the landscape structure. And then through extension, how does this affect community processes, community assembly, and beta diversity? So some previous work in Dr. Trexler's lab is focused on directionality and activity rates of animals in these different deep water and shallow water habitats. And a good example is, especially with things like mosquito fish and flagfish, as the water levels start to increase, you see this big spike in activity, and that's as the animals are immigrating out or moving out of these deep water areas. And then as water levels stabilize, you tend to see big decrease in activity. That's just as animals are kind of milling around for a couple months, eating And then as the water levels start to go down again, you see a big increase in activity as everything's moving back to the deep water areas. And so in this particular study, this is an area in the central Everglades. It's known as the Gap. It's about 2 kilometers wide, 40 kilometers long. And the only deep water habitat in this area is this black line here is actually a deep water canal. It's about 15 feet deep, the L67C. And then immediately to the east, there's an earthen levee, so animals can't move to the east. And to the west, it's just a shallow marsh that's of uniform depth for those 2 kilometers. The light areas are sloughs, open water sloughs dominated by spike rush, lily pads, things like that. And the dark areas are sawgrass, which animals can still move through, though the vegetation is considerably denser. There's 3 sites that I set up immediately adjacent to the canal, CB sites, and they're as close to the canal as I could be without being in the canal. And then 150 to 300 meters in, I have these marsh sites, the UB sites, trying to set them up as a trans site, though there are pretty big differences between each one of these sites. And we sampled this area, well, the sampling's still ongoing, but for the purposes of this project, sampling occurred 10 times between 2010 and 2012, and I'm just trying to get a good representative mix of the change in water levels, not so much set on calendar days, but as water levels are going down and rising. So to determine density and a measure of the small fish community, we use these meter squared throw traps, which are just open-ended boxes you go out and throw them. And then to determine directionality and movement, we have these roughly 24-foot wide drift fences set out over 24 hours, and there are minnow traps arrayed in the middle in the different cardinal directions. Go back and collect them, and from that you can get a measure of directionality over a 24-hour period through catch per unit effort. So in this particular case, these are mosquito fish. There's a lot of animals that you'd be catching in the east trap, a few in the north trap. So you'd say that there's roughly a west-southwest movement in this particular area. So why do we think that that changes in movement? Why am I asking this question? If you look at the small fish density, we do see that there is very little difference between the nearer canal sites and the marsh sites for density. There's a little bit greater density in the near canal sites, but nothing striking. And same with the drift fence CPUE catch. Again, very few differences over the course of time. But if we look at an NMDS plot of the community, we do see the blue is the canal sites, red are marsh sites. You do see some pretty stark differences in where the communities lie, especially with throw trap. And Adonis reveals the significance of this as well. And if you look at which fishes are contributing the most to the abundancies of these different sampling types, you do see different species are promoting this change. So not only is density and catch per unit effort similar, you are seeing differences in communities between these 2 different sites. So that was just a long way of saying communities are different, and why are we thinking that they're different? So to look at this at a more fine scale, I picked out 5 species that I catch a lot of, and they kind of fall into 3 groups. We've got eastern mosquito fish and dollar sunfish, which are quick movers and fast to recover during periods of dry down. So especially eastern mosquito fish have really aren't impacted much by a dry down at all. At the other end of the spectrum, we have golden top minnow and bluefin killies, and they're slow movers, and they take a while to recover from a strong dry down. Especially the bluefin killifish, it can take several years after a bad drought for them to go back to their original densities. And then in the middle, we've got the chubby little flagfish, who is kind of an in-between, where they're slow movers, but they respond really well to a dry down. They're really good at getting through the really tough conditions, and often their densities are highest right after a dry down. And so we went into this with some a priori hypotheses. Because of the way that the canal was arrayed, it's very convenient that we can look at the directions as either towards or away from the canal. So just thinking about this in terms of adaptive responses, as the water level is going up, we would predict that animals are moving away from the canal to access these new areas. Water levels are going down, they're moving back and then speed will increase during marsh water recession, which again is just an adaptive response that makes sense. All right, really quickly, golden topmail, one of the slow movers or slow to respond. I'll just get you set up here on these graphs because this is what it's going to look like for the next 6 slides. On the y-axis, there's the predicted probability of direction. On the x-axis, there's the water depth rate of change, decreasing, stable, or increasing for every one of these. And then the dashed line is towards, solid line is away. And what we do see here is kind of what we would expect for this near canal site. So as water level is decreasing, they're moving back towards the canal, and then you kind of lose significance as water level is increasing. Switching over to the marsh, things are a little more muddied, but generally what you see again is as water levels are increasing, you're seeing movement away from the canal. Going on to the mosquito fish, one of the rapid movers, quick to respond to a dry down. And this is very similar to what we see in the dollar sunfish. Really tight directionality, though it's almost always away from the canal at all times, regardless of water going up or down. And you see similar results in marsh conditions too. So both near canal and marsh, these animals are just always moving at a pretty good speed away from the canal. And then moving on to the flagfish, again, somewhere in the middle, where as water levels are decreasing, he's still moving away from the canal. And similar conditions for the marsh too, although in the case of the marsh, they are moving away from the canal as water levels are increasing, which agrees some with their a priori hypotheses. I didn't talk much about speed, mostly because there are very few significant differences in speed between habitat type or water depth change. Though it is important to note that speed was significantly greater during the periods of greatest drawdown, which would have been March and April of 2011 and 2012, and that's when water levels are dropping really fast. And at that point, especially in 2011, if things weren't leaving the marsh, they were going to die. So again, that fits nicely with our hypotheses. So just a quick summary. The slow dispersers, the guys slow to recover. There's some directionality of movement, often towards the canal during falling water levels. Fast dispersers, quick to recover, often very significant directionality, but always away from the canal. And then, well, let's go through that. Speeds increase during periods of greatest water direction. And so this just fits into the, I also have some electroshocking data and large fish movement data, and then also small macroinvertebrate data giving me, hopefully will be a nice picture of a whole community, size structure, community structure, and how these animals change based on proximity to the canal, especially in the context of a big restoration project that's going on out there. And I'm sorry I went over. Lots of people to thank, including some very awesome, dedicated technicians. We have time for a few questions. Really quick questions.
Yeah. So I know that a lot of these organisms take refuge in solution holes, things like that. And is there really any effect of the availability of these solution holes, which are refugia during the dry down season? So if they're moving away from the canal, you don't expect that they would do that during the dry down, but maybe they're moving towards these solution holes. Is that, have you considered that at all?
In this area, I've only found one gator hole. So
oh, really? Okay.
If it's not the canal, that is the source. And I think what's happening is, well, I think what's happening is that these things that are quickly passing, they're just blasted across the landscape.
Yeah, makes sense.
Happens to us if they find it. Slow movers are more cautious. That's just my personal opinion.
One quick question from you. So those fish that are constantly always moving away from the canals, would that feel like kind of like a mass effects thing?
Yes.
Where the canal is the source? And
I think that's how I consider it as a beta diversity.